In early October 1968, Mexico City was on edge. The long-awaited Olympic Games were due to begin in just a matter of weeks, and throughout the city a specialized Olympia battalion of the military had been ordered by President Diaz-Ordaz to roam the city and crack down on any signs of dissent. The people had many reasons for dissent. Decades of economic growth had not done enough to improve the lives of average Mexicans, and this was accompanied by a political climate of suppressing workers' rights. Mexico is no stranger to people's revolutionary movements, and in 1968 it was the students of UNAM who ended up with their lives on the line in defiance of the government. On October 2nd, roughly 10,000 students assembled peacefully in the huge open quad in the center of Mexico's biggest and most famous university, the National Autonomous University of Mexico, UNAM. Students had formed a national strike council to resist the government, and on this day members of the council and thousands of interested bystanders were listening to speeches as they began to hear helicopters circling overhead. Within half an hour, flares were being shot from the helicopters, and thousands of soldiers had begun to encircle the campus. When the smoke cleared, 28 students had been killed, and hundreds wounded. And across the city, in a quiet living room, a small child pushed himself up from the floor and went over to turn on the radio. He quickly shifted away from the news playing the horrors of the day's events and found a music station playing last year's hit, Good Vibrations by the Beach Boys. The song sent an electric thrill through the little boy, and he got up and started dancing. 13 years later, that child was a young man, Luis Herrera Estrella, and he was sitting and listening to a speech at that same university that those years ago had been the site of a bloodbath. But instead of listening to speeches on worker solidarity, he was listening to a Belgian plant scientist, Professor Marc von Montague. The professor had been in Jamaica attending a conference on plasmids, the circular DNA molecules that had become the hottest topic in science due to the creation of recombinant engineered plasmids in a lab in the San Francisco Bay Area just a few years prior. Herrera Estrella sat in the auditorium listening to Professor von Montague talk about the peculiar plasmids of a species of gall-forming bacteria, Agrobacterium tumefaciens. These bacteria naturally infect plants and have a unique molecular machinery to transfer their plasmid DNA, not from bacterial cell to bacterial cell as is common for bacteria. No, these agrobacterium cells can transfer plasmids directly into the cell and into the nucleus of a plant as they infect it. The bacterial DNA actually forces its way into the fortress of the cell's genome, cuts open a place for itself somewhere randomly in the genome, and integrates, getting stitched into the host's own genetic material. There, the bacterial genes appear to the plant like any other genes, and they are transcribed and translated to produce proteins that force the plant cell to become a factory of nutrients for the bacteria, dividing rapidly to form large masses of undifferentiated tissue called a gall, the plant equivalent of a cancerous tumor. This was all a revelation for Herrera Estrella, who had just finished a master's in genetics at the Center for Research and Advanced Studies in Mexico City, but who was not a plant scientist and did not know about the weird world of plant galls. But because of his background in genetics, he immediately saw the potential that Professor von Montague also saw in this system. If we could harness the newly created tools of recombinant DNA technology developed in the San Francisco labs and use it to modify the special plasmids of Agrobacterium, then you could hijack the system as a human tool. As the bacterium uses its DNA to hijack the plant genome, and humans with our advanced scientific tools could hijack the hijacker, turning this naturally evolved platform for gall formation into a way to genetically modify plants at will. Writing 29 years later in an essay in the Plant Biotechnology Journal, Professor Herrera Estrella recalled a moment of déjà vu. Hearing Professor von Montague talk sent that same bolt of energy through him as when he had listened to Good Vibrations as a 4-year-old. An energy full of impatience to dance or to change the world, and he knew he had to act on it. Just 2 years later, after listening to the speech at UNAM, Herrera Estrella had achieved exactly what he had imagined in that lecture hall, harnessing the natural plasmid system of Agrobacterium tumefaciens to create genetically engineered plant cells. And in doing so, he created the path forward for thousands of plant scientists to come, and ushered in the era of genetically modified crops. Welcome to Modified. I'm your host, Dr. Orlando Delange, ex-plant scientist and current science teacher. This podcast is my attempt to guide you through the science of GMOs and hopefully just enough of the history and the social context to give you a path forward in your own thinking. And if all else fails, I hope you'll enjoy some of the surprising stories and fascinating facts in the world of genetically modified plants. This is episode 10, How to Make a GMO Plant. Over the middle of the 20th century, genes had gone from hidden properties of plants to a very tangible raw material for those interested in shaping the traits of crops. In episode 11, we'll pick up from episode 9 and talk about the commercial world of biotechnology and the launch of the world's first GMO crop. By the early 1980s, there were tools to see genes, to read sequence, and to physically isolate and rearrange pieces of DNA. It took no time at all for scientists interested in both basic plant biology and in plant breeding to try to physically insert pieces of DNA into places they hadn't been before. For those interested in the basic biology, this could be a very powerful way to answer questions about the function of genes and connect that back to the biological processes. And that might only involve moving DNA that all comes from the same original source. So for example, say you have a mutant plant that doesn't grow fine root hairs. You have a few suspicions about the genes that are responsible. Basically, you hypothesize that there is some gene in this plant species that encodes a protein that is important for making root hairs, and that your plant has a mutation in that gene that leads to a different, non-functional protein that can't make root hairs. If you have a few leads on what this gene is, you can use a combination of PCR, molecular cloning, and transgenics to insert functional copies of each gene into the plant genome and test whether the new transgenic plants are now able to make root hairs. And if so, that is a really strong confirmation of your hypothesis. And that approach started to get used more and more over the end of the 20th century, eventually becoming one of the most common methods used in studies of plant biology. You can also use the same overall suite of methods to find mutations in genes you're interested in and see what happens to the plants with these mutations. And at the same time as basic researchers were developing this reliable set of molecular genetic methods for basic research, applied scientists and professional plant breeders were trying to make the most of all these technologies to develop new crops through transgenics. The appeal of transgenics to plant breeders are manifold, and we'll explore them in more detail in Season 2 of the podcast. But a few of the top attractions are: number 1, you can create totally novel traits, unlike anything else on the market, because you aren't reliant on just shuffling around traits that already exist in populations of the crop. You can draw from genes in any organism on Earth, or even create totally new genes that have never existed in nature, and then stick them into your crop. Both these approaches were taken right from the start in the 1980s. And the big advantage of doing something totally novel is that you'll have a unique product to put on the market, not just one of several very similar crop varieties sold by other companies. And that's going to give you a commercial advantage. Number 2, you can, in theory, save a lot of time. Plant breeding is a slow process of using sexual reproduction to keep mixing together sets of alleles until you finally end up with the combination that you're looking for. Directly adding in the genes you want is often, but not always, faster. Number 3, since you are creating something that doesn't exist in nature, you have more options to patent it, which brings economic benefits, and we'll explore that aspect a lot more in Season 2 of this podcast. But in addition, I don't think we should discount just the excitement of a new technology driving forward interest in plant transgenics. Consider the abundance of AI companies today attracting venture capital money. In the 1980s, a suite of totally new plant breeding companies sprung up, hoping to capitalize on the promise of plant transgenics, and we'll learn more about them in the coming episodes. But first, this whole episode will be about unpacking the science that makes it possible to create GMO crops in the first place. I won't go back over how to create recombinant plasmids by sticking together DNA pieces. If you're interested in that, you can go back and listen to episode 9 if you haven't already. Instead, let's assume we know how to easily create totally new DNA molecules that take DNA from different sources and combine it together. How do you get that new recombinant DNA inside of a plant genome? There are two main challenges to creating a GMO plant. And since the early days of plant transgenics in the 1980s through to today, both of these challenges remain. Challenge 1, how to get the DNA into the genome of a plant cell. Challenge 2, how to take that plant cell and turn it into a whole plant. Of these, Challenge 2 is actually the one that plant scientists and plant breeders have been working on for a lot longer, for well over a century. For many plants, we have robust methods developed that allow us to basically break them into a bunch of cells, and then take those cells, turn them into stem cells, which are capable of regenerating whole plants. This is never an easy trick to pull off, requiring specialist tools and expert technicians. But for most economically and scientifically important plants, methods have been developed. Still, for many plants that challenge at taking a plant, converting it into individual free-floating cells, and then converting those cells back into a whole plant can still be elusive. We can bundle all these processes together under the term cell culture. The first challenge, how to get DNA into a plant cell, is something that we have two methods for, both invented in the 1980s. The agrobacterium method described in The Cold Open, and the gene gun, which is really what it sounds like, developed just a few years later in 1986. At least those are the two most widely used methods that have been most successful. And just like cell culture, exactly how to use these methods to get DNA into plant cells requires optimization for each target plant species. But how do you actually make a transgenic plant? Actually, isolating and sequencing the DNA is the easy part. Today it's extremely easy. An undergraduate with a few plant molecular biology courses under their belt could easily design a new gene, put in an order from one of the many companies to chemically synthesize billions of copies of their DNA sequence, and send them via post in a few days to the undergraduate researcher. In the 1980s, it wasn't that simple, but it also was something that a relatively small team of scientists could achieve in about a year, basically going from basic idea to a tube of designer DNA pieces ready for action. What can take longer is getting them into your plant of interest, recovering healthy baby plants from the process, and screening thousands of offspring for ones that have the correct DNA insertion without any unintended side effects. Plant cell culture was developed over the first half of the 20th century. In 1902, Austrian scientist Gottlieb Hebeland first isolated plant cells and grew them successfully for some time as simply cells in a petri dish. And he envisioned that with more research it might be possible to induce those cells to divide and produce a whole new plant. Interestingly, he was in the middle of a multi-generational group of famous Haberland scientists and agriculturists. His father, Friedrich Haberland, played a massive part in introducing soybeans to Europe, and his son, Ludwig, carried out pioneering reproductive biology research that paved the way for the development of the human birth control pill. Like any other profession, science can run down generations, since children will have easy access to information about scientific careers and how to pursue them, and likely they will have encouraging parents facilitating their scientific education and career development. Haberland made significant contributions, but his ideas did not come from nowhere. There are agricultural science texts from 6th century China that describe the process of grafting pear tree branches onto apple trees. This technique is a core technology of tree fruit agriculture, and so it's been long known that plant cells have a developmental plasticity far beyond animals, that even small pieces of an adult plant can grow again into a full adult, given the right conditions. If you've taken a cutting from a houseplant and put it into water, you've probably observed this for yourself. Many plants, particularly tropical plants, will readily generate new organs and whole new plants from cuttings, as long as those cuttings contain a meristem, which, if you remember from episode 4, is the name for the pocket of stem cells found at the end of every plant root and shoot, and also found at regular intervals on stems at points known as nodes. And those nodal meristems allow the formation of side branches and side roots, but also whole new plants if the conditions are right. And those plants which do not naturally do this trick can be made to do so with the right application of nutrients and hormones. That's cytokinins, auxins, and our old friends, the gibberellins. I mentioned just previously that plant cell culture was pioneered at the start of the 20th century. As the name suggests, plant cell culture is the ability to break down plant tissues into free-floating cells and have those cells continue to divide and grow basically as a soup, similar to the way bacteria and yeast grow. And generally, these cell suspensions are grown in darkness, providing sugar in place of sunlight. Remember, the trick of photosynthesis is to use sunlight to turn carbon dioxide and water into sugar, but you can also just provide the plant cells directly with sugar. For most plant genetic transformation methods, you do not need or want to work with plant cell cultures, per se, but the development of cell culture techniques in the 20th century went hand in hand with the development of a wide range of what we call plant tissue culture techniques, meaning taking small pieces of plants, not necessarily single cells, and then forcing them to regenerate into whole plants. But really it's one related set of methods, variations on the exact right culture media and hormone mixes to get the desired result, whether you're working with individual cells or small pieces of tissue. Whether you're going for plant tissue or cell culture, you have to be very good at aseptic technique. That is basically a set of tools and practices to keep unwanted microbes out of your work. As I mentioned, plant tissue culture relies on providing the plant cells with pieces of sugar and growth media, generally in a reasonably warm and humid environment to avoid desiccation. All of that makes for a perfect environment for many bacteria and fungi. There are some chemical additives that can suppress microbial growth, but skilled aseptic technicians are essential for a successful plant transgenics operation. For many plant transformation methods, the ideal form of plant matter to work with is what we call a callus, plural calli. Calli are clumps of undifferentiated cells. To prepare calli, you can take any rapidly dividing plant tissue, pieces of growing shoots, roots, or pieces of the developing plant embryo, or germinated seedlings are all common inputs. Then little pieces of this tissue, called explants, are placed onto a growth medium. Not just any old growth medium, the exact set of nutrients, minerals, and vitamins, and crucially plant hormones, will be tested extensively to find the right balance, which will differ for each plant and each tissue. But when you get the right balance, use effective aseptic technique, and provide the right environmental conditions, then the calli will start to form and grow over a period of days. And you do not always need calli as the input for DNA delivery. Sometimes the explants with an active meristem are a good enough input. But assuming that you are working with a plant species that does need calli for the process, what next? Well, at this point, imagine a petri dish with some pale-coloured jelly, that's the growth medium, and on it a set of tiny pale green or white knobbly clumps, each about the size of a matchstick head or a bit bigger. And now these are the calli, and you can take them, isolate them, and keep growing them and isolating them until they become a much looser mush of cells, and ready to turn into a free-floating cell culture. Or you can take the calli earlier on in their development and use them as the input for plant transgenics, using methods that I'll explain in just a minute. And once you've delivered the DNA, you transfer the calli to a new growth medium, because whereas at first we wanted to take the plant tissue and turn it into cells, or at least undifferentiated clumps of cells, now we want to take those cells and turn them into a plant. And so, just like for the first growth medium, the second medium, the regeneration medium, will have gone through its own careful process of optimization. And at this stage, many of those calli will die before they make it back into producing a whole plant you can transplant to soil, but by now, after decades, robust methods have been developed for many species, so there's definitely still a lot of skill and equipment needed, but it is robust and reliable. In fact, if all of this sounds like fun, and you'd like to try your own hand at plant tissue culture and cell culture, then you can actually get equipment, materials, and training at reasonable prices from a couple of US-based companies, Phytotech Labs and Plant Cell Technology. And you don't need to get all the way to making calli and cell cultures to have fun with plant growth media and hormones. You can use plant tissue culture techniques to regenerate whole plants from tissue explants as an efficient way to propagate your beloved houseplants and share them with all your friends. Now let's move on to DNA delivery. Can you picture the little petri dish with plant cell blobs called calli ready to receive recombinant DNA? How do you actually do that? I'll describe 3 methods. The first one I already described in the cold open, so let's just refresh that. Delivery via the vector Bacterium agrobacterium tumefaciens. This was the first technique developed and remains the most common because it's so easy. All you have to do is assemble your genes of interest into a plasmid, flanked by a special set of DNA sequences that are recognized by the agrobacterium DNA delivery machinery. If you want the fancy name to impress your friends with, it's the type 4 secretion system, the set of proteins that find these DNA sequences, cut them out of the plasmid DNA, and shuttle them directly into the host cell, and then cut and paste them into the host genome at a random position. Basically, scientists took the naturally evolved transfer plasmids of agrobacterium, removed all the bits that aren't useful to humans or are actually harmful to the plant host, and now you have a useful tool to deliver whatever genes you want into the plant host. The main downside of this approach, and it's a pretty important one when it comes to thinking about GMO crops, is that the DNA gets inserted randomly into the plant genome. That could be into the middle of a gene that actually has some important role in the plant, so there is room for unintended side effects. The inserted genes, along with the flanking regions from the agrobacterium plasmid, are called the tDNA. So part of the process of creating a transgenic plant using this method is checking where the tDNA landed in the plant genome, and just checking the overall phenotype of the plant to ensure that nothing important got messed up in the process. Another downside of tDNA is the tDNA, meaning the flanking sequences from the agrobacterium that have no real value from a human perspective once they've done their job of getting the interesting and useful DNA into the target plant genome. They don't do anything bad. If you've eaten food in America, then likely you have eaten plenty of tDNA. It doesn't encode any proteins, so it's just DNA, no different chemically from other DNA. What's so great about this method is that you basically get the bacteria to do all the hard work. They naturally infect plants, multiplying in the plant tissue and delivering the tDNA into as many plant cells as possible. So the researchers can just let the bacteria do all that. Unfortunately, the bacteria can do too good of a job and actually damage and sicken the fragile calli in the process. So part of the trick with this method is getting the conditions just right for a good infection, but one that doesn't do lasting damage and can be easily cleared with physical rinsing and with antibiotics to kill off any lingering agrobacterium cells. This job is made easier by the fact that the bacterial strains used in labs have been domesticated to make them unable to function effectively as pathogens, just good enough to deliver DNA and then get out of the way. So that's the Agrobacterium tumefaciens tDNA method. Another very common method is biolistics. It's not as common in research labs because it requires specialist equipment instead of just growing some bacteria, which is very cheap and easy, but it is used to some extent in research labs and has been extensively used in developing transgenic crops. Partly because some plants, particularly cereal crops, do not play nicely with agrobacterium, and biolistics can be a more successful approach. So what is biolistics? As you might guess, it's the combination of bio and ballistics, and the other name is a gene gun. The first model was indeed a modified air rifle, but instead of shooting pellets a few millimeters across, it shoots nanoscopic particles of gold coated with DNA. Using a helium propellant, the tiny gold particles are small enough that when they get shot into calli, they can enter the individual nuclei of cells. Then, once the DNA is inside the nucleus, the genes will be expressed. If the DNA is just floating around the nucleus, this gene expression will be transient. Free-floating DNA not part of a chromosome is always eventually destroyed. But by chance, the tiny gold particles can hit the chromosome body, tearing a small hole, and during repair, the DNA on the gold particle can be integrated at the breakpoint. Biolistics is not a method I have direct experience with, and if anyone is interested to learn more, then I'd be happy to put in some extra research on the method and how it works. If you want, submit a question about it to our upcoming second installment of Your Questions Answered. The final method I want to mention is protoplast transfection. A protoplast is a plant cell without its cell wall. Now, this is not any way for a plant cell to live. They hate it. They can't live very long in this state, and you have to be very careful to keep the protoplast in a liquid suspension with just the right balance of salts to stop the cells taking on so much water that they burst, but also to stop them from desiccating. And they will start regrowing their cell walls just a few hours after you remove them. And that's done by the applications of cell wall digesting enzymes, either to calli or to little pieces of normal plant tissue or to cells in suspension. The advantage of protoplasts is that you add some helper chemicals and a whole bunch of plasmid DNA to them, and they will take up DNA into their cells, and some of it will make it into the nucleus. And then just like with biolistics, the genes on that DNA will be expressed transiently. But unlike biolistics, there is no mechanism in this process to tear holes in the chromosome, so it's very, very unlikely that an individual plasmid added via protoplast transfection will find its way to getting integrated into the plant genome. As I'll explain in a minute, even very rare events can be found, like the proverbial needle in the haystack, if you have the right selection method. But actually, this feature of protoplast transfection, its transience, is being harnessed as a major benefit for what many consider to be a major future direction for plant breeding: non-transgenic gene editing using CRISPR-Cas9. We'll pick this up as the final episode of this season, but basically CRISPR-Cas9 is a relatively recent new tool for cutting and modifying DNA, and this allows you to go in and tweak the existing DNA in the plant genome, rather than what we think of as a GMO crop when we're adding in DNA from another organism, what we call a transgene, resulting in a plant that has DNA from some other organism integrated into its own genome, and we call such a plant transgenic. If you use protoplast transfection to add the genes that encode the CRISPR-Cas9 machinery, it can be in the cells long enough to edit the plant's own DNA, and then it will be removed completely in the process of turning those cells into a whole plant. And so what you have now is a transgene-free plant, and many in the industry argue that what you can achieve in this approach is basically the same sorts of DNA changes that are achieved using non-genetic engineering-based plant breeding approaches, and therefore that plants developed using this method should not be regulated and subjected to labeling laws in the same way as GMO plants. And that battle is currently playing out in the courts and legislatures around the world right now. So now we've talked through how you can break into plant cells, deliver DNA into them, and turn them back into a whole plant. There's just a little problem, and we'll round out this episode by exploring it. How do you know which recovered plants actually have your gene in them? The DNA delivery processes are all happening at the nanoscale, far too small to see. All you know is that you took some cells, you gave them some of the foreign DNA, and that you turned some of those cells back into whole plants. But you don't know which of those regenerated plants actually got the DNA. One answer is to use PCR, that method invented in 1983 that allows you to use short molecular probes called primers to basically look for specific DNA sequences and amplify them in vitro, in tiny tubes. You can check for successful amplification easily by running the reaction product through a jello-like agarose gel and staining for DNA to see a band of DNA at the expected size, and that tells you that the template DNA you were looking for was indeed in the original sample. And that is always part of the process for creating a GMO plant, checking for DNA insertion using PCR. These days, that would be followed with sequencing the whole genome of the specific recovered plant to see exactly where the recombinant DNA ended up in the plant genome, and that first became logistically feasible in the 2010s. Still, even with those tools to check for the presence of the recombinant DNA, the transgene as it's known, you will have to screen an awful lot of plants to find the very few that actually have the transgene. So in most cases, the whole process is helped along by including an accessory transgene alongside the thing that you're actually interested in, bundled together into the plasmid DNA using the delivery of the DNA into the plant cell. And this accessory gene is often a gene that encodes a protein that destroys a chemical herbicide that would otherwise kill the plant cell. This is the easiest approach because by definition, only the plants that have the transgenes will be recovered because they're the only ones that can survive application of the herbicide. And because the herbicide resistance gene will be delivered as a packet, physically bundled with the gene you're more interested in, then you can be very confident that a herbicide-resistant plant recovered after DNA delivery will have any other genes delivered alongside it as part of the same original plasmid that you designed in the lab for this purpose. Another approach is to make the plants that got the DNA, what we call successful transformants, more visible. For example, by including a gene that produces a protein that glows green when you shine UV light on it. So you just shine UV light over the whole plate of Cali after DNA delivery, and you can quickly see which ones glow green and pick them out. Those are the successful transformants. So that's it. All you do is culture plant tissue to produce Cali, deliver DNA via one of multiple methods, and then screen the surviving Cali to identify successful transformants. That's the basic workflow for every plant and for every variety of every plant, but the precise details of how you do this will be optimized in each case if you want to be successful. And then along the way, you will need specialist equipment, chemicals, and a lot of expertise, particularly in aseptic technique, basically keeping unwanted bacteria and molds out of the process. Like I said earlier in the episode, if that all sounds very exciting to you and you'd like to invest some time and money in learning how to do all this for yourself and create your own plant transgenic setup in your own home, well, you could. First, you'll need maybe 100 square feet that you're happy to set aside and devote to this. And then you'll want to go to companies like Plant Cell Technology or Phytotech Labs to start getting materials and training. And if you don't have the space, and you'd love to be able to learn and practice alongside other interested and interesting folks, then see if there's a community biolab near you. These are not very common, but some cities do have non-profit molecular biology laboratories that anyone can join and use to do projects. I know, because I used to help run one in Seattle. In about a month, I'll be doing another Your Questions Answered episode, which is exactly what it sounds like. This will be the final one of the season, and it's the perfect opportunity if anything in this episode or recent episodes has gotten you curious to learn more about recombinant DNA technology and genetic engineering. Send your questions to orlando@modifiedpod.com. That's ORLANDO @modifiedpod, all one word, .com. Before we finish with our 5 take-home messages for today, I want to thank a few key references for this episode. I want to acknowledge My Journey into the Birth of Plant Transgenesis and its Impact on Modern Plant Biology, an article in Plant Biotechnology Journal by Luis Herrera Estrella from 2020. And I want to acknowledge Trevor A. Thorpe's article, A History of Plant Cell Culture from 2013. Those articles, along with other important sources for this episode, are available at the podcast website, which is modifiedpod.com. That is modifiedpod.com. Please, if you're enjoying this podcast, do subscribe and leave a review. That makes it far more likely that other people will also be able to find it. And of course, tell a friend. I really appreciate it. It's been wonderful to see more and more people listening each week, and I'd love to reach an even wider audience. So let's end with your 5 take-home messages. Number 1. A transgene is DNA from a different organism. When integrated into a target organism, you create a transgenic organism. The process of adding the new DNA is genetic transformation. Number 2. To create transgenic plants, you usually have to start by preparing tiny blobs of undifferentiated cells called calli. Number 3. Agrobacterium tumefaciens is a plant pathogen that has been turned into a tool for DNA delivery into plant cells. Number 4. Gene guns are another tool used to deliver DNA and create GMO crops. Number 5. To create a transgenic plant after DNA delivery, you need the right conditions, and these usually include a way to easily screen for successful transformants. That's all for this episode. Bye!